we're back with our Edexcel GCSE Biology Paper 1 predictions for 2025. Last year, we came extremely close to predicting almost every single topic correctly. This year, we've once again analysed all the trends and patterns to bring you the most likely topics for your exam. Our predicted papers include exam-style questions designed to match the real thing. Mark schemes so you know exactly how to score top marks, and new for 2025, we've got free video walkthroughs showing you how to structure your answers in the way examiners expect. If you're looking for even more support, our masterclasses include access to our Revision Accelerator course over the school holidays and our live exam prep sessions the night before your exam for a final confidence boost too. We've done all we can to make your revision and study easier, but of course, as always, remember to revise everything to be fully prepared. Now, let's get into our 2025 predicted topics for Edexcel GCSE Biology Paper 1. We're going to start with separate higher, then combined higher, separate foundation, and then combined foundation. Be sure to skip to your relevant section using the timestamps. First up, like we said, for separate higher, revise cell structure and specialised cells. Revise the key differences between prokaryotic and eukaryotic cells, including features such as the nucleus and organelles. You also need to know how specialised cells are adapted for their functions, including the sperm cells, which have a tail for swimming and lots of mitochondria for energy. Nerve cells, which are long with branched connections to transmit signals quickly. Finally, make sure you know the functions of cell organelles, such as the nucleus, mitochondria, and ribosomes. Next, enzymes are a major focus too. You need to understand the lock and key theory, which explains how enzymes work. Brush up on factors affecting enzyme activity as well, including temperature and pH, because extreme values can denature enzymes, and substrate concentration, as increasing concentration speeds up reactions until saturation. Also, be prepared to recall the role of digestive enzymes such as amylase, which breaks down starch into glucose, protease, which breaks down proteins into amino acids, and lipase, which breaks down fats into fatty acids and glycerol. Another possible topic is the brain and imaging techniques. You need to know the functions of key brain parts. Cerebrum, responsible for thinking, memory, and emotions. The cerebellum, which controls balance and coordination, and the medulla, which regulates heartbeat and breathing. In terms of imaging, make sure you understand MRI scans, which detect brain damage and diseases, and CT scans, which provide 3D images of the brain structure. Next, brush up on microscopy. Make sure you can compare light microscopes versus electron microscopes, including their resolution, magnification, and what they reveal. You also need to be confident using the magnification formula. That's magnification equals image size divided by actual size. And another key skill is understanding how to repair a microscope slide, including the importance of staining cells to make structures more visible. Our next topic is inheritance, sex determination, and sex linkage. You should be able to explain how sex is determined using chromosomes, XX for females and XY for males, how sex-linked conditions such as colour blindness and haemophilia are passed down through the X chromosome, and how to use Punnett squares to predict inheritance of traits. You'll also want to revisit fossils and evolution. Make sure you can describe how fossils form, including mineral replacement and preservation in ice, amber or peat. Our next topic is selective breeding. It is used in farming to produce high-yield crops and disease-resistant animals, and PhETs to breed animals with desirable traits. However, inbreeding can reduce genetic variation and cause health issues. Next, be sure to revisit cloning. Types of cloning include tissue culture for growing plants from a few cells, embryo transplants where embryos are split to create clones, and adult cell cloning, which was used to create Dolly the sheep. Cloning has benefits in medicine and conservation, but also reduces genetic diversity. Our next topic is sexual and asexual reproduction. Make sure you understand the differences between sexual reproduction, which creates genetic variation but it's a slower process, and asexual reproduction, which produces identical offspring quickly. Examples include bacteria, fungi reproducing asexually, while some plants can reproduce both sexually and asexually, such as strawberries using runners. Another topic to brush up on is aseptic technique used to prevent contamination when growing bacteria. Key steps include sterilising equipment, such as flaming the inoculating loop, sealing the petri dish to prevent unwanted bacteria growth, and incubating at 25 degrees Celsius to prevent harmful bacteria from growing. Nearly there, revise drug trials too. You need to know the stages of drug testing. So, 1, preclinical trials tested on cells and animals, 2, clinical trials tested on humans in different phases, and 3, double blind trials where neither the doctor nor patient knows who gets the real drug or a placebo. Double blind trials are important because they remove bias and increase reliability. Next, we have monoclonal antibodies, which are made by fusing a B lymphocyte with a tumour cell to create a hybridoma. This hybridoma produces identical antibodies for medical use. Uses include treating cancer, pregnancy tests, and detecting diseases. Finally, malaria is also a key topic. It's caused by a protist called Plasmodium, which is spread by mosquitoes. To prevent malaria, strategies include using mosquito nets, draining stagnant water, and taking anti-malarial drugs. These are our higher separate predicted topics for 2025, but remember, the exam could include anything, so it's essential to revise everything. Here are some final tips. Practice past papers or our predicted papers to improve exam technique. Use mark schemes to understand how examiners award points. And then, obviously, if you need any extra support, join our masterclasses where we provide structured revision, boosters, and live exam prep sessions the night before your exam. Most importantly, take care of yourself. Exams are important, but your mental well-being matters too. So take breaks, stay focused, and remember you are more than just your grades. Let's now move on to higher combined. So this section is for our 2025 predictions for Edexcel GCSE Combined Science for Higher Paper 1. These are the topics that are likely to appear, but remember, revise everything to be fully prepared. First up, revise cell structure and specialised cells. With prokaryotic and eukaryotic cells, you need to understand the key differences, such as the presence of a nucleus and organelles. Then, with specialised cells and their adaptations, know that a sperm cell has a tail for movement and mitochondria for energy. A nerve cell is long with branches to transmit signals quickly. And finally here, just look into the functions of organelles. Get to know the roles of the nucleus, which stores DNA, mitochondria, which produces energy, and ribosomes, which synthesizes proteins. Next, let's talk about enzymes. Look into the lock and key theory. Enzymes have a specific shape that fits their substrate perfectly. Consider factors affecting enzyme activity too, so things like temperature and pH, because if these are too high or too low, it can denature enzymes, and then substrate concentration too, which increases reaction rate until enzymes become saturated. And then finally, here look into digestive enzymes. Amylase breaks down starch into sugars, protease breaks down protein into amino acids, and lipase breaks down fats into fatty acids and glycerol. Microscopy is the next topic. Look into light versus electron microscopes. Know their differences in resolution, magnification, and what they reveal. Know how to calculate magnification. The calculation is magnification equals image size divided by actual size, and of course, know how to prepare a slide. Staining cells helps structures become clearer. You'll then want to revisit inheritance and sex determination. Where chromosomes are concerned, you have XX for female and XY for male. The Y chromosome triggers those male characteristics. You've then got dominant versus recessive alleles, where dominant traits only need one copy to be expressed, and just finally here, know that Punnett squares can be used to predict the inheritance of traits. Moving on to gene therapy, you should know what it is, that being the process of replacing faulty genes with healthy ones to treat genetic diseases. Examples include treating cystic fibrosis by inserting a normal CFTR gene, and it's used in cancer treatment to target cancer cells. There's challenges and risks here though. It's difficult to insert genes in the right place, and it's expensive and still being developed. Our next predictions are based on fossils and human evolution. Know how fossils form, so make sure you can describe how fossils form, including mineral replacement and their preservation in ice, amber, or peat. Next, onto selective breeding. You need to know how it works. Humans will select parents with desirable traits. Know its uses too. It's used in farming, typically for producing high yield crops and disease resistant livestock, and it's used for pets, so for example, breeding dogs with gentle temperaments. Finally, know the risks. So inbreeding can cause genetic disorders, and reduced genetic variation makes species more vulnerable to disease. We also think that you should look into drug development. You should revise the stages of drug testing, that's preclinical trials where they're tested on cells and animals, then clinical trials, phases 1 to 3, where they're tested on humans, and then there's double blind trials where some patients receive the real drug while others get a placebo. Blind trials are important because they remove bias and increase reliability. The final topic we think may come up is malaria. Know that malaria is caused by a protist, Plasmodium, which is spread by mosquitoes. Symptoms include fever, chills, headache, and muscle pain, and prevention methods include using mosquito nets, draining stagnant water, and taking anti-malarial drugs. Okay, so those are our combined higher predictions for 2025. Just as some final advice for you guys, practice past papers and check mark schemes to understand how examiners award points. You'll of course find a more detailed breakdown of our topics and what they might look like as a question in our predicted papers too. If you need extra support, join our masterclasses. We offer revision boosters and live exam prep sessions the night before to help you feel confident, but most importantly, take care of yourself. Exams are important, but your mental health matters, so take breaks, eat well, and remember that grades do not define you. Let's now move on to separate foundation predictions. We have carefully selected key topics for Edexcel GCSE separate science, but remember to revise everything to ensure you are fully prepared. First up, we think that you should revise specialised cells. Cells have unique adaptations to help them perform their specific functions. For example, sperm cells have a tail that allows them to swim and contain a high number of mitochondria to generate the energy required for movement. Nerve cells are long and have connections that enable them to transmit electrical signals quickly. Next, revise microscopes and magnification, including practical techniques. Light microscopes have lower magnification and can view living cells, whereas electron microscopes provide much greater magnification and detail, though they can only be used for dead specimens. Make sure you also know the formula for calculating magnification, which is magnification equals image size divided by actual size. To prepare a slide for viewing, a thin specimen should be placed on a slide and a stain such as iodine can be used to highlight structures. And of course, the cover slip must be lowered carefully to avoid trapping air bubbles, which can interfere with visibility. Next, you want to look into DNA and inheritance, including sex determination and inherited diseases. DNA stores genetic information and is found in chromosomes within the nucleus of cells. The sex of an individual is determined by their chromosomes, so females have XX chromosomes, while males have XY chromosomes, with the Y chromosome responsible for triggering those male characteristics. You should know that some diseases are inherited through genes. Okay, so next we have fossils and evolution. Fossils are formed when hard parts of organisms, such as bones and shells, are gradually replaced by minerals. Some fossils are preserved in ice, amber, or peat. These remains provide a valuable evidence of how species have changed and evolved over time. Evolution occurs through natural selection. Mutations cause variation within a population, and organisms with beneficial adaptations are more likely to survive and reproduce. Over many generations, these traits become more common, leading to evolutionary changes in species. Now, the next topic we think will come up is the nervous system. The nervous system enables the body to detect and respond to stimuli. When a stimulus, such as touching a hot surface, is detected by receptors, a signal travels through sensory neurons to a relay neuron. This message is then transmitted to a motor neuron, which sends a signal to an effector, such as a muscle, prompting it to move the hand away from the heat. Reflex actions like these are fast and automatic, helping to protect the body from harm. Now, your next topic is selective breeding and genetic modification. Selective breeding involves humans choosing parents with desirable traits to breed together. This technique is commonly used in farming to produce larger crops, disease-resistant animals, and pets with specific features. However, one of the risks of selective breeding is inbreeding, which can lead to health problems due to a lack of genetic diversity. Now, genetic modification, or GM, involves altering an organism's DNA to give it new traits. For example, scientists can modify crops to make them resistant to pests. The benefits of genetic modification include increased yield and improved resistance to disease, but concerns remain regarding ethical implications and potential long-term effects on the environment. Our next predicted topic is food tests. Food tests are used to identify different nutrients in food samples. Benedict's test detects sugars and turns blue to a brick red if sugar is present. Iodine test is used for starch, turning from brown to orange to blue and black if starch is detected. PhET test identifies proteins changing from blue to purple when protein is present. Our final predicted topic that we think you should revisit is plant diseases and aseptic technique. Plants can suffer from various diseases caused by fungi, bacteria, viruses, and pests. Examples include tobacco mosaic virus, that's TMV, rose black spot, and aphid infestations. Symptoms of plant diseases include discoloration, spots, and stunted growth, and they can be identified using testing kits or laboratory analysis. Aseptic techniques are essential in microbiology to prevent contamination of bacterial cultures. Key steps include sterilising equipment, so for example, flaming the inoculating loop, sealing the petri dish to prevent the entry of unwanted bacteria, and incubating at 25 degrees Celsius, which prevents the growth of harmful bacteria. Okay, those are our separate foundation topics. While these topics are predicted to appear in the exam, it is essential to revise everything to ensure full prep. Practicing past papers and our predicted papers will help you become familiar with the types of questions that may come up and improve your exam technique. For additional support, consider joining our masterclasses too. We offer revision boosters, live exam preparation sessions the night before your exam, and plenty more to help build your confidence. Most importantly, take care of yourself. Exam stress is real, but your grades do not define your worth. Take breaks, eat well, and believe in yourself. You've got this. Okay, finally, here are our combined foundation predictions too. These are key topics that we think may come up in the exam, so make sure you are confident with them. Okay, so first up, we think that you should revise specialised cells. Cells in the body are adapted to perform specific functions. So, for example, sperm cells have a tail that allows them to swim toward the egg, and they contain a large number of mitochondria to provide the energy needed for movement. Nerd cells are long and have branches that help transmit electrical signals quickly. Specialised cells are essential because different jobs in the body require different structures to function effectively. Next, revisit microscopes and magnification, including practical techniques. There are two main types of microscopes. You've got light microscopes and electron microscopes. Electron microscopes have a much higher magnification and resolution, allowing scientists to see smaller details within cells. The magnification of an image can be calculated using the formula magnification equals image size divided by actual size. Now, when preparing a slide for viewing under a microscope, a thin specimen should be placed on a slide and a stain such as iodine can be added to highlight structures. The cover slip should be lowered carefully to prevent air bubbles from forming, which can interfere with the clarity of the image too. Next up, revisit DNA and inheritance, including sex determination and inherited diseases. Know that DNA carries genetic information and is found in the nucleus of cells stored within chromosomes. The sex of an individual is determined by their chromosomes. Females have XX chromosomes, while males have XY chromosomes, with the Y chromosome triggering male characteristics. Some diseases are inherited through genes. Cystic fibrosis is caused by a faulty recessive allele, meaning that an individual needs to inherit two copies of the faulty gene to develop the condition. In contrast, polydactyly is caused by a dominant allele, meaning that only one copy of the faulty gene is needed for the trait to appear. Our next predicted topic is fossils and evolution. Fossils are formed when the hard parts of organisms, such as bones and shells, are gradually replaced by minerals over time. In some cases, fossils can also be preserved in ice, amber, or peat. Fossils are important because they provide evidence of how life has changed and evolved over millions of years. Evolution occurs through natural selection. Mutations cause variation within a population, and organisms with the best adaptations are more likely to survive and reproduce. Over generations, these traits become more common, leading to evolutionary changes in species. Now, nearly there, our next topic is the nervous system. The nervous system allows the body to detect and respond to stimuli. For example, when touching something hot, the stimulus is detected by a receptor and a signal travels through the sensory neuron to a relay neuron. The message is then passed to a motor neuron, which sends a signal to an effector, such as a muscle, causing it to move the hand away from the heat. Reflex actions like these are important because they are fast and automatic, helping to protect the body from harm. Okay, finally, we also think you should revise selective breeding and genetic modification. Selective breeding involves humans choosing parents with desirable traits to breed together. This technique is commonly used in farming to produce crops that grow faster or animals that are resistant to disease. It's also used to breed pets with specific features. However, one of the risks of selective breeding is inbreeding, which can lead to health problems due to a lack of genetic diversity. Genetic modification, or GM, involves changing an organism's DNA to give it new traits. So, for example, scientists can modify crops to make them resistant to pests. The benefits of genetic modification include improved yields and increased resistance to disease, but there are concerns about its long term effects on the environment and ethical issues surrounding the practice as well. Okay, so those are all of our predictions. These topics are predicted to appear in the exam, but it's obviously important, like I keep saying, to revise everything you're worried about to ensure you are fully prepared. Practicing past papers and our predicted papers will help you become more familiar with the types of questions that may come up and improve your exam technique as well. Like I said before, we offer revision boosters and live exam prep sessions the night before the exam to help you feel ready and confident. And you can sign up to all this and more through our masterclasses, which now have a pay monthly option. Finally, remember to take care of yourself. Exam stress is real, but your grades do not define you. If you're trying your absolute best, you're doing enough. Now, good luck.